Hello friends. Welcome to my YouTube channel. Eager for you. Please like, subscribe, and press bell icon to get notified my educational videos.
Hello friends, in this video I am going to discuss one of the important topics in plant tissue culture, that is micropropagation. So, contents are 1. Definition 2. Stages of micropropagation 3. Types of micropropagation 4. Advantages of micropropagation and 5. Disadvantages of micropropagation.
Friends, micropropagation is defined as the rapid in vitro clonal propagation of plants. Clonal propagation means multiplication of genetically identical individuals by asexual reproduction. Here, you can see how an explant of a mother plant is developed into clones. Friends, Georges Morel was the first to start use of tissue culture technique for micropropagation of occurred in 1960 and now applied to several plants. Micropropagation provides a sufficient number of plantlets from a stock plant which does not produce seeds, or does not respond well to vegetative production.
Stages of micropropagation. Morashich in the year 1974 outlined 3 major stages. Toshio Morashich, most widely known for his efforts in creating the MS medium. These 3 stages are 1. Establishment stage 2. Proliferation stage and 3. Rooting and hardening. These 3 stages involves 1. Selection of plant material where healthy mother plant, free from disease and insect pests, is selected, from which explants are excised. 2. Establishment of aseptic culture, where explants are surface sterilized and transferred to artificial nutrient media aseptically. 3. Multiplication of shoots. Here, shoots are multiplied by subculturing. 4. Shoot elongation. Here, the multiplied shoots are elongated. 5. Root induction. The shoots are transferred to the rooting media where roots are induced. 6. Hardening or acclimatization. Here, the tissue-cultured plantlets are transferred to hardening chambers like polyhouse and later transferred to soil. Sometimes 2 more stages are added, one at start and another at last. These stages are renamed as Stage 0, Stage 1, Stage 2, Stage 3, and Stage 4. Micropropagation and plant regeneration can be grouped into 1. Axillary bud proliferation, which can further be divided into meristem shoot tip culture and bud culture. In this diagram, from a rosette plant, meristem or shoot tip is isolated and cultured on artificial media, then shoot is induced. Now, with the help of cytokinin, axillary branching is developed, followed by rooting of axillary shoots. From elongated plant, meristem or nodal portion is isolated and cultured in artificial media, then shoot is induced. Now with the help of cytokinin, axillary branching is developed, followed by rooting of axillary shoots. Second type of micropropagation is organogenesis, which is defined as the process of formation of monopolar structure, that is shoot or root, either directly from explant, called direct organogenesis, or through coluse derived from explant, called indirect organogenesis. Here diagram shows the explants derived from a healthy mother plant when cultured on artificial media. Shoots are induced either directly or through coluse and cell suspension culture. Further, roots are induced and whole plant is regenerated. Third type is somatic embryogenesis, which means the formation of bipolar structure, that is embryoid, either directly from explant, called direct somatic embryogenesis, or through coluse derived from explant, called indirect somatic embryogenesis. The diagram shows the induction of somatic embryos either directly from the explants or from the collusion or cell suspension culture. Further, shoots and roots are induced, followed by regeneration of whole plantlet. Friends, I always try to limit the length of my videos up to 10 minutes, so due to time constraint, all these methods will be explained separately in further coming videos.
Advantages of micropropagation. Here we will discuss why this method is so important in comparison to the conventional methods. 1. The small amount of plant tissue is needed to produce millions of plants. 2. Commercial mass propagation of plants can be done in relatively less growing space and time. 3. Period of quarantine is reduced. It provides fast international exchange of plant material without the risk of disease introduction. 4. It provides year-round nursery. We can develop nursery plants throughout the year irrespective of seasonal variations. 5. It helps in the production of disease-free plants. Generally, meristem shoot tip cultures are employed to develop pathogen-free plants. 6. Seed production. Micropropagation is suitable for seed production in some plants through axillary bud proliferation method. This is also required to multiply plants which produce seeds in uneconomical amounts or when plants are sterile and do not produce viable seeds. 7. Germplasm storage. The germplasm stocks can be maintained for several years using this technique. 8. Artificial seeds. Through somatic embryogenesis, production of synthetic artificial seed is becoming popular nowadays.
Disadvantages of micropropagation. 1. Expensive requirements and trained manpower needed. For some micropropagation techniques, expensive equipment, sophisticated facilities and trained manpower are needed. 2. Contamination can cause very high losses in a short time. During the course of micropropagation, several slow-growing microorganisms contaminate and grow in cultures. The microbial infection can be controlled by addition of antibiotics or fungicides. 3. Problem of genetic variability may be pronounced in some varieties. When micropropagation is carried out through shoot tip cultures, genetic variability is very low. However, use of adventitious shoots is often associated with pronounced genetic variability. 4. Vitrification. During the course of repeated in vitro shoot multiplication, the cultures exhibit water-soaked or almost translucent leaves. Such leaves cannot grow and even may die. This phenomenon is referred to as vitrification. This can be prevented by increasing the agar concentration in the medium. However, increased agar concentration reduces the growth rate of tissues.